Hello everyone, my name is立典. Today I'm giving you a quick preview of a paper titled Human iPSC-based breast cancer model identifies S100P-dependent cancer stemness induced by BRCA1 mutation. This research presents a new way to study breast cancer, discovering a new gene that can be useful as a drug target. First, let's look at the challenge in breast cancer research. Breast cancer is a leading cause of death for women, but our understanding of its cellular and molecular basis is still limited. This is mainly because we lack good human-based research models that can simulate the disease development. The BRCA1 mutation is strongly linked to triple-negative breast cancer, which is difficult to treat. So finding a way to study this is critical for developing new diagnostic and therapeutic targets. To address this, the researchers focused on developing a new model using induced pluripotent stem cells, or iPSCs. The main challenge was to get functional memory cells from these iPSCs. Based on the knowledge of how mammary glands develop, they created a new protocol to differentiate iPSCs into memory-like cells. As you can see from the diagram, they took cells from a patient with familial breast cancer and reprogrammed them into iPSCs. Then they used their new protocol to differentiate these cells into memory cells. This allowed them to study how BRCA1 mutation affects human breast cells directly. Finally, these memory cells were transplanted into mice to see if they would form tumors, which they did. The key finding is that the iPSC model successfully mimicked the characteristics of human breast cancer cells. These cells show similar genetic changes and the ability to form tumors. When transplanted into mice, only the BRCA1 mutant cells formed tumors, and they look just like primary human breast tumors. Next, the researchers identified a specific gene called S100P. This gene acts as an oncogene downstream of the mutated BRCA1 gene. They found that S100P promotes cancer stemness, which is the ability of cancer cells to self-renew and spread. This discovery is significant because it suggests that S100P could be a promising new drug target for breast cancer. Figure C shows a heat map of gene expression, highlighting the high expression of S100P in the patient-derived cells. Figure D shows the result of inhibiting S100P, demonstrating how this can reduce growth of cancer cells. To conclude, this study establishes a new and promising system for studying the mechanism of breast cancer. The iPSC-based model can be used to find new therapeutic targets and screen for potential drugs. Thank you for listening.